think about, just like we did in class, think about the ways that they are similar and different, and then maybe we'll start class on Wednesday with sharing of information about this, okay? So, we'll skip the answer, and then there's another one. What ways are secondary active transporters like or unlike uniporters, which are passive transporters? What are some of the similarities, and what are some of the differences? So, do those two similarity difference questions as homework. So let's do this one together. Because this one is related to the question 2 that you've worked on. So I'm not going to give you the answer to question 2 because I want you to work together on it on Wednesday. But this is an opportunity to have a sort of a different kind of conversation about it. If the sodium potassium ATPase is inhibited by 50%, what will happen to the intracellular concentrations of the following? So if the sodium pump is inhibited, what will happen to the intracellular sodium concentration? So show me as a clipper. A, increase, B, decrease, C, no change, D, I could argue either way. About the direction that the sodium potassium normally pumps, the sodium potassium pump normally pumps sodium, and then what would happen if you inhibited it by a large margin, by half? Okay, I'm seeing mostly correct answers. The sodium concentration inside is low because the pump is pumping it out. If the pump is not pumping it out as well, the concentration will increase because of those leaks, channels, other transporters. The sodium concentration would tend to increase because the pump is not working as effectively to pump it out. Okay, what about potassium? Right, so now, same logic, okay? We're going to skip calcium because that's the problem. Let's go to glucose. If the sodium potassium ATPase is inhibited by a large amount, 50%, that's probably fatal, but by a large amount, what would happen to the glucose concentration? Okay, so I'm seeing all kinds of color here. I'm seeing A, B, C's, and D's. So somebody who says the glucose concentration, so I see a lot of B's. Somebody who argued for the B answer. Somebody who chose B. Tell me how you came up with B. So
the co-transporter wouldn't have the sodium gradient as strong to bring glucose into the cell.
Okay, so Sophie's remembering there is a sodium glucose co-transporter that uses the sodium gradient to pump glucose into the cell. And so if the sodium gradient is impaired, because now the sodium inside is higher than it should be, so the gradient between outside and inside is collapsed or impaired. Less sodium gradient, less energy for glucose pumping in. Okay? Somebody who argued for A, I saw a couple of A's, or C's. I saw a couple of those too. Give me your answer. Tell me how you came up with that. Okay, you probably can tell that that A is a correct answer, but I think the best answer is D. You could argue either way. So make an argument for how glucose would increase. Let's see, wait, did we argue for, no, we argued for decrease. So we already did this one. Make an argument for how glucose could increase if you inhibited the sodium-potassium ATPase. Where am I going with that? Abby?
Maybe less ATP is being used, and so less glucose is being metabolized.
Exactly. So could you hear over there? No. So if the ATPase is inhibited by 50%, the cell is using less ATP and the sodium potassium ATPase is using a large amount of the ATP that the cell produces through glycolysis, Krebs cycle, electron transport chain, ATP synthase. So the cell is not using as much ATP, so it's not doing glycolysis, et cetera, as much. So the glucose concentration could increase because it's not consuming glucose as fast. Was that your idea? So that's true. I mean, that could be possible. So it could decrease because there's less demand for ATP, or it could increase because of the lower gradient for sodium glucose cotransport. So why did I do this? I did this because sometimes I will throw you a curveball like this, and you could argue either answer, and the quality of your answer, I mean, the amount of credit you get is whether you think about it, or whether you can argue sort of both sides of the argument. There are going to be times when, well, if I look at it this way, I get to this answer, and if I look at it this way, I get to this answer, and they're both going on, and neither of them is wrong. So maybe it's no change because you could come up with no change because of both of these happening at exactly equal effectiveness. Anyway, the point is sometimes it's going to be tricky. Okay, let's, for the next 45 minutes or so, turn our attention to signaling, both chemical and electrical. So some of this is going to be, all of this is going to be reviewed from self-risk. So we're talking about receptors, a signaling molecule interacting with a receptor that has selectivity for the signaling molecule. Here is a cartoon from your A&P textbook that shows a hormone or a neurotransmitter or a signaling molecule binding to a receptor, interacting with a G protein. This should ring bells. The G protein interacting with a effector molecule and generating a second messenger. So there are things about this figure that I dislike intensely, but I'm not going to go into it. But one of them that drives me nuts is the idea that the receptor is big and the G protein is small. That's just not true. The G protein is almost is bigger than the receptor. What is missing? I just wanted to see, what do you know about G proteins? What is missing? So this shows the alpha subunit dissociating from the receptor and from the beta gamma complex. That's good. What's missing
The ATP and ADP conversion.
here? The what?
The ATP to ADP conversion.
That's what's going on here. This is ATP to cyclic AMP.
It doesn't,
What happens here?
it's not activated.
Isn't it GTP?
GTP.
Okay, yeah.
You say ATP, or
GTP,
I'm not gonna give you full
I was
credit.
thinking there was an energy molecule in there.
GTP binds here, and that's what triggers, that's what activates the alpha subunit, GTP binding. So here is a slide from Cell-Phys. Let's walk through it. This is a cartoon, obviously, but it's better. So this figure right up here is just setting everything up. Let's make sure we understand the cartooniness of this cartoon. What are these gray slabs? I think I heard the answer. The bilayer, the membrane. It's the surface membrane. Oh, it's right there. Yeah, the surface membrane. So that's another point. Sometimes I'll ask you a question and the answer's up on the screen. So don't assume that I know that I've asked a stupid question. Okay, so here's the receptor protein and here's the G protein. What are these zigzags? Anchoring. What kind of anchor anchors the G protein? It's a lipid. So this is a fatty acid, sort of a lipid tail that is anchoring this G protein to the membrane. Remember we talked about membrane proteins, so transmembrane proteins and lipid-linked integral membrane proteins. This is a good example of one of those lipid-linked integral membrane proteins. Okay, somebody used your words to tell me what's going on here. What is this illustrating right here? Two words. One begins with a D and the other begins with a B. Diffusion and binding. Right? I won't. I beat this one enough, so I won't do that again. Okay. What are these red blasty things supposed to illustrate? Activation, a conformational change. So the binding of the signal molecule, the hormone or the receptor or neurotransmitter or other signaling molecule, binding, conformational change, activation of the receptor. What happens to the alpha subunit? What happens to the alpha subunit as a result of the conformational change of the receptor?
Releases GDP.
Okay, so yeah, it releases GDP, but why? There's two words, and I'll beat this one enough too. Conformational change of the alpha subunit. Lowers the affinity for GDP. GDP dissociates. What is this arrow illustrating? GTP binds to the alpha subunit. So receptor conformational change lowers the affinity for GDP, but raises the affinity for GTP. GTP binds, or GDP unbinds and GTP binds. This should be ringing a bell from Cell-Phys a little bit. What is illustrated here in this activated situation? What does the binding of GTP do? Separation? Go ahead, Remy, tell me, what is separating from what?
The alpha beta complexes separate.
Okay, so the alpha is separating from beta gamma, and both of them are separating from the receptor. And what are these blasty lines supposed to be illustrating?
It flips on the screen.
Hmm?
The screen changed.
Oh. This pointer has a super sensitive button. What are these blasty lines trying to communicate to you cartoony wise? Conformational change, and what is this doing to this? Activated, right? So now we have an activated alpha, and we have an activated beta gamma. Both of those are signaling entities. Oh, so here's the story that I was trying to get you to tell. Diffusion and binding, conformational change of the receptor, GPCR, the receptor. Conformational change of the G protein, alpha. GDP dissociates, GTP associates. Dissociation of beta gamma from alpha, and both dissociate from the receptor. And both the GTP bound alpha and the liberated beta gamma can go on to activate other things. Again, this is a story that we told a lot in Cell-Phys. I want you to reintegrate that into your conscious memory. So here's the cartoon from your cell A&P textbook. We have a GTP activated alpha that is activating an effector. What is this effector doing?
In the GS case,
In this case,
like the kinase cascade.
yeah, so have we activated? Yes, so we're going to get to a kinase cascade. But what is this protein doing? I mean, it's obvious. This is a stupid question. Yeah, it's turning ATP into cyclic AMP. It's a cyclase. It's turning a linear ATP into a cyclic molecule. And cyclic AMP is a messenger inside the cells. So, but this is interesting, right? ATP, which you think of as an energy molecule, is now being turned into a signaling molecule. So ATP has multiple uses, not just energy. And then cyclic AMP can activate, what are the three initials for this protein right here that we learned about in Cell-Phys? PKA, protein kinase A. Cyclic AMP dependent protein kinase. What do kinases do? Phosphorylate substrates and change them. So here is ATP. Let's do just a tiny bit of chemistry. Here is the structure of ATP. Adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. Adenylyl cyclase converts ATP into cyclic monophosphate. There's the cyclic. There's the cyclic. This is the signaling molecule that's active. In almost all cases, if we turn something on, we have to have a way to turn it off. If we don't turn it off, usually that means cancer. Uncontrolled something. So there are enzymes in your cells called phosphodiesterases that cut cyclic AMP into just ordinary AMP, which is not a signaling molecule. AMP is the substrate that can get converted with phosphorylation back into ATP. Remember, the T stands for triphosphate, and M stands for monophosphate, and so monophosphate is different from triphosphate. this is not an energy molecule and it's not a very good signaling molecule. This is a signaling molecule, but not a very good energy molecule. This is actually both. Okay, so let's do a problem. There are receptors in your body that are coupled to GI. So let me, I'm going a little fast because that's the nature of the beast. What's the name of this G protein that activates adenylyl cyclase? GS. So this is the alpha subunit of GS. There are other G proteins coupled to other receptors that inhibit adenylyl cyclase, and those are called GI. Very similar, but instead of activating adenylyl cyclase, they inhibit adenylyl cyclase. So, you have receptors in your brain and elsewhere in your body that are activated by ligands, signaling molecules. For example, adenosine. Adenosine is a signaling molecule. It activates receptors that are coupled to GI. When you drink coffee, it is an inhibitor of those receptors, but it is also an inhibitor of phosphodiesterase. It's an inhibitor of this enzyme. So caffeine inhibits a receptor and it inhibits this enzyme. What does caffeine do to your intracellular cyclic AMP levels? Show me an answer and be prepared to defend it. This one's easy once you get it, but it's hard until then, like all problems. Okay, somebody who chose, well, somebody defend your answer. Tell me your answer and defend it. No?
So, caffeine, so GI is like an inhibitory G protein, and adenylyl cyclase is an activator. So if it's an antagonist to that one, so if they're antagonists, they work opposite to one another. And that means if caffeine is, and can you go back to the question?
Yeah.
Yeah, so caffeine is an antagonist of AO1Rs, which are the GI proteins, and it's also an inhibitor of phosphodiesterase, which is an inhibitory GI. So caffeine increases cyclic AMP in the intracellular.
So you're saying that the answer is A, it increases it. Somebody want to argue against that?
I mean, I kind of agree with him, but I'd say it was C because the A1R that's coupled inhibits the AC, which means there's going to be less cyclic AMP. But the caffeine inhibits that A1R, so there's going to be more cyclic AMP.
Okay,
also,
good. So let's unpack that. Do we believe that? So if the G protein is inhibiting adenylyl cyclase, but we inhibit the inhibitor, we inhibit the production of the inhibitor because we're inhibiting the receptor. So if we inhibit the receptor that's making the inhibitor, then this enzyme will tend to be activated by other things, right? So we're going to activate adenylyl cyclase by inhibiting the inhibitor, and so that would make more cyclic AMP. So right there we say, okay, by that mechanism, cyclic AMP would go up a little bit. Go ahead.
And then, so it said it inhibits the phosphodiesterase too.
Right.
So that, actually, I think I'm going against my answer now. Could you go back to the question real quick? So, actually, I think it might just be A, now that I'm looking at it
Okay.
closer.
So thinking about it more, you started agreement, but then you were saying, wait, no, that's not right, but then so you see where Nahum was going? By inhibiting an inhibitor, we're producing more cyclic AMP. And then by inhibiting the breakdown of cyclic AMP, we're allowing more cyclic AMP to accumulate. So both of these effects are important for the increase in cyclic AMP when you drink coffee. And that stimulates your brain. The increase in cyclic AMP is activating certain neurons. It's activating your heart. Your heart beats harder, beats faster. You've experienced it. You drink coffee and your blood pressure goes up. We're going to be talking about that a lot in the next week or so. One of the key points is inhibiting an inhibitor can act like a stimulator, because there's almost always something going on to allow, there's something stimulating this and something inhibiting it. And if we inhibit the inhibitor, the something that's stimulating it is now sort of out of balance and it's acting more. Okay, that's the key point. Here is a slide of the GQ story, which I am not going to talk about because it is your question, the second question too, on your problem set. So this should be a resource. This slide should be a resource for the question 2 on page 2. And this slide is your resource. This is the first one we've been talking about. This is your resource for question 4. So that's homework and further discussion in the in the Zoom breakout room. We'll talk about this associated with your problem solving. And I'm going to skip this. Well, I'm just going to say that a kinase cascade, I think it was Vernusha used the word kinase cascade. No? Somebody's used the word cascade. Activating a kinase, which activate enzymes, there's multiplication. And so if we activate one receptor, we might activate 1 million downstream effector molecules because of multiplication of these in the cascade. We'll talk about that when we get to some specific examples. I'm going to skip that one. I'm going to skip that one because so let's do this one as part of the Zoom breakout problem. Okay? So I'll remind you on Wednesday that this should be one of the problems. Maybe I'll post it separately so it's right there. Okay? Because I want to get to electrical signal. Okay, so this is going to This is really important because we can't understand the heartbeat without understanding electrical signaling. But it's 3:20, and I've had you here for, since 1:40. So I get a sense of, saturation. I'm torn. I'm gonna look at my schedule and see whether I could pick up some time later so that I could, not do this now. Because I'm concerned that if I do this now, it won't